Still on chapter 21. I know this is a long chapter. We have been talking about the adaptive defense system. And the adaptive defense system includes humoral immunity and cellular immunity. And cellular immunity is carried out by the T cells. And so we are going to focus on cellular immune response for this particular talk. So T cells provide defense against intracellular antigens. Remember, the cellular immunity has cellular targets, right? So example would be cells infected with viruses or bacteria, cancerous or abnormal cells, or foreign cells, which means like transplanted cells. Some T cells directly kill cells, and others will release chemicals that will help to regulate the immune response. T cells are more complex than B cells in both classification and function, and there are two populations of T cells with different what are called cell differentiation glycoprotein receptors that are displayed on their surface. And the two of them are CD4 cells and CD8 cells. CD4 cells usually become helper T cells, and those helper T cells are going to go off and activate B cells and other T cells, as well as macrophages, and they also help to direct the adaptive immune response. Now, some of these CD4 cells do become regulatory T cells, which can help moderate the immune response, and they can also become memory T cells. So CD4 cells become either helper T cells, regulatory T cells, or memory T cells. CD8 cells become cytotoxic T cells, which are going to be capable of destroying cells that have that foreign antigen that corresponds with that particular T cell. And they can also become memory T cells. Now, once a cell is called, a T cell is called either a helper T cell or a cytotoxic T cell or a regulatory T cell, those are all activated T cells. If the T cell is still naive, it's just simply called CD4 or CD8 cell. And so what we have here is we have these, this is just showing immature lymphocyte, and it matures either into a CD4 or a CD8. If it becomes a CD4, once an antigen is presented to it, it becomes activated and it differentiates into the effector cells. And the effector cells are either the helper T cells or the regulatory T cells. And again, it can also become memory cells. CD8 cells, oops, sorry about that. Same thing. Antigen is presented to it, it becomes activated, it differentiates into either memory cells or these effector cells, which are, in the case of the CD8 cells, are going to be cytotoxic T cells. So again, T cells only respond to processed fragments of antigens that are displayed on the surfaces of those antigen-presenting cells by the major histocompatibility complex, or MHC protein. And this antigen presentation is vital for the activation of naive T cells and normal functioning of effector T cells. There are two classes of MHC proteins. There's a class 1 MHC protein. This is displayed on all cells except for red blood cells. And class 2 MHC proteins are displayed by antigen-presenting cells. And remember, our three big players there are going to be dendritic cells, macrophages, and B cells. And both types are synthesized in the endoplasmic reticulum and bind to peptide fragments. Class 1 MHC is crucial for CD8 activation. Remember, CD8 cells are going to become cytotoxic T cells. So what it's going to do is it's going to act as an antigen holder, and the MHC protein is going to form the self part that the T cell recognizes. And that goes way back to, I believe, the last talk where we talked about the selection process, both positive and negative selection. And it has to recognize the self part as well as, then I need to move on to this part now. Then we also have it informs the cytotoxic T cells of microorganisms hiding in the cells. And the negative selection part comes in, where it ignores the displayed self-antigens. So this part would be positive selection, and this part would be the negative selection. Class 2 MHC proteins are recognized by helper T cells. This is just, again, a nice little table showing you the role of MHC proteins in cellular immunity, with the class that's displayed by each cell, what it's recognized by, the message that it's sending, a lot of good information in this table. All right, MHC restriction. CD4 and CD8 cells have different requirements for MHC proteins that present antigens to them. The CD4 cells that become T helper cells are restricted to binding only to class 2 MHC proteins, typically on antigen-presenting surfaces. And CD8 cells become cytotoxic T cells, and they're restricted to binding to only class 1 MHC proteins found on every cell surface, including antigen-presenting cells. And then once the cytotoxic T cell becomes activated, it seeks out the same antigen on the class 1 MHC protein on any cell. So now we're going to talk about activation and differentiation of T cells. And they, again, are only activated when an antigen is presented to them. And then activation is a two-step process. Antigen binding and co-stimulation both occur on the surface of the same antigen-presenting cell, and both are required for clonal selection of the T cell. So with antigen presenting, the T cell receptors bind to the antigen MHC complex on antigen presenting cell surface. So again, we go back to remembering the fact that we have antigen presenting cells and they have this MHC complex on them, or this MHC protein on them. And once the antigen presenting cell has engulfed the whatever it is that it's trying, the antigen, and it presents it, it's going to present it on this MHC, this antigen MHC complex on its surface. The T cell antigen receptor is then going to bind to that on the antigen presenting cell surface. The TCR must perform double recognition by recognizing both the MHC and the foreign antigen it displays. And then binding of the TCR to the complex is going to trigger a number of intracellular signaling pathways that are going to start the activation of the T cell. Then we have co-stimulation. Complete T cell activation is going to require that T cell to bind to one or more co-stimulatory signals on the surface of the antigen presenting cell. Without this co-stimulation, something called anergy occurs, where the T cells become tolerant to that antigen, they're unable to divide, and they do not secrete cytokines. And we're going to talk about cytokines very, very briefly here in a moment. This is a two-step process that safeguards against unwanted T cell activation. Then we have proliferation and differentiation. Once the T cells become activated, they proliferate in response to cytokines, and they differentiate and perform the function according to whatever their T cell class is. So if they're a T helper cell, they're going to go do things that T helper cells do. If they're a cytotoxic T cell, they're going to go do things that cytotoxic T cells do, like kill virus-infected cells, for example. And a helper T cell may do something like go activate a B cell. Primary T cell response peaks within a week, and then apoptosis begins after about a week, and somewhere between a week and 30 days. And then activated T cells are hazardous because of the fact that they do produce a large amount of inflammatory cytokines, and this could result in hyperplasia or cancer if not cleared from the system. And hyperplasia is like the tissue is enlarging, more tissue is being added. Memory T cells can remain and mediate secondary responses. So we have antigen presentation. Again, the antigen is engulfed by the antigen presenting cell. The little antigen fragment is coupled with this MHC protein. and then it's going to bind. We have double recognition, antigen binding, and then co-stimulatory, and then we have clone formation. And so this is going to go off and form its clones, and these guys are going to do, again, whatever their specific job is. So helper T cells, again, may go and activate B cells. Cytokines are just chemical messengers that, chemical messengers of the immune system. They mediate cell development, differentiation, and responses in the immune system. They include interferons, and interleukin. But I didn't ask you about any specific ones. I did leave this table in here in the event that you wanted to look up what specific interferons and interleukins do, but you do not have to know this table or what, again, any of the specific cytokines do. And that's all for this talk. And there's no bonus on this one. There will be bonus on the next one, though.